In LabKey Lim Suite, you can track the lineage of your samples, from the source of where your samples originate, such as a patient or study, to their child samples, such as aliquots or derivatives. To configure your lineage, navigate to your sample type, click Manage, click Edit Sample Type Design. Add a parent if the sample type may be derived from a different sample type. Add a source type if the sample may come from something that isn't a sample, like a patient. You can set these as required as needed. Click Finish Updating Sample Type. When you register a new sample, the lineage settings that you just created will now appear with a drop-down of available samples or sources that are already registered. You can also create lineage between samples by selecting the parent sample or source. Click Derive, then click Aliquot Selected, Derive from Selected, or Pool Selected. An aliquot sample will always be the same sample type as the parent sample and generally has the same metadata associated with it. For example, an aliquot would be if I am splitting a blood tube into smaller tubes. A derivative means that the parent sample and the child sample have different sets of metadata and are often different sample types. For example, a DNA sample is derived from a blood sample. A pooled sample means I am combining part of or all of multiple parent samples into a singular sample. When you are creating a child sample, you will be prompted to update the parent sample status. Select the status you desire. If the status has a consumed status behavior, you will also be prompted to remove the sample from storage. Enter in the child sample's data, then finish creating the sample. To view the lineage of that sample or its source, navigate to that specific sample or source's page and click on Lineage. This will provide a visual representation of how a sample or source is related to other samples. You'll be able to filter through the status of the samples that are displayed here. You can click anywhere on the diagram to bring up more metadata at each level. For sources that have many samples, click the Samples page to go to the grid view of every sample derived from that source. If you need to correct a mistake or need to update the lineage of a sample, click the checkbox by that sample, click Edit Lineage, and make the updates that are needed.